share with you now the overview of immune response. Okay. Okay, yeah. So here what has happened is the overview of immune response you can see. So what are the different types of antigens that you can encounter? It could be a foreign protein, okay, it could be a virus, and then it could be an antigen. Let us see this. When we are talking about foreign proteins, it could be a part of the virus or it could be a part of the bacteria. So if it is not the full scale bacteria, then we shall call it foreign protein. Okay. For example, in order to produce an antibody, usually what we do is we inject ovalbumin into a mouse. Okay. So when we inject ovalbumin in the mouse, what happens is, I'll show you the structure of this. Vibrio. Now see, this is a Vibrio Antigen which my students are working to develop into something. Anyway, just see the structure of the antigen. It is not the whole Vibrio altogether, but a small part of it. Okay. You can see the screen, right? All of you. Okay. Now this is the tertiary structure. We will convert into a tertiary structure. Okay. And color it by secondary structure. Okay. Now this is a surface protein present in on Vibrio surface on the polysaccharide. The peptidoglycan layer is there. Now on the layer there are some embedded proteins. Okay, so it's one protein, very very important and very a good candidate for making it into a vaccine. So what exactly happens? This is the structure of this. We can generate a 3D structure so that you can understand. Okay, so this is a small part of the whole surface of the whole peptidoglycan of the Vibrio. It's a small part, so it's a typical subunit vaccine. So if you inject it into a vertebrate animal, what it will do is it will be considered as an antigen and not as a a bacteria, and accordingly immune response will be initiated. So same is true for viral antigens, cancer antigens as well. Okay. So what will happen is, these are the major types of antigens which will enter into the system. Okay. Now whenever a bacteria enters, here you see what exactly happens. The antigen is internalized by an antigen presenting cell. Okay. And a part of the whole antigen is being presented by a thing called class 2 MHC molecules. Okay. Now here you see carefully, it was a bacteria. Now bacterial antigen has been inserted and digested internally by a thing called lysosome mediated degradation. Now what happens in all animals, lysosome mediated degradation is very very important. So what it does is it disintegrates the whole antigen and breaks down into pieces and then present it through MHC2 molecules. Then you can say, sir, why MHC2 molecules? Now MHC2 molecules will be presenting these bacterial antigens to helper T-lymphocytes. Okay, so I have not spoken here. I have to tell you before that. Okay, just give me a little time. So I have told you about T lymphocytes. Okay, I think many of you already know it, but I'll once again repeat, if in case some students cannot remember, or so what exactly happens is T lymphocytes are further having two different classes. Okay, so what are the classes of T lymphocytes? One is called TH cells. Okay, very important also, TH cells. Okay, and another is called TC cells. Okay, now what are these TC cells? I'll discuss. So TC cells are nothing but cytotoxic T lymphocyte. As you can remember, the name also suggests cytotoxic means it is toxic for the cells. Okay, which cells? Altered self cells. Please remember. Now what is an altered self cell? Whenever a foreign virus has caused some infection in a host cell, so what will happen? The host cell will change its character, isn't it? At that point of time, an infected cell is converted into an altered self cell. So whenever any infection takes place inside a cell, the cell converts into an altered self cell, not self, altered cell. So helper cytotoxic T lymphocytes will disintegrate or destroy the altered self cells. Okay, but what about the TH cells? Helper T cell, as the name suggests, helper T cells means these are the cells which will help all kinds of immune response related cells to flourish in their number, capacity and everything. Okay, so I'll give you one more example that what is the role of helper T cell. It's very important also to understand. So whenever, as I have said, that this antigen, this B lymphocyte is virgin. Okay, so whenever an antigen is encountered, B lymphocyte will convert into two things. Remember this. A B lymphocyte will convert into two things. What it will convert into? It will convert into, just one second. It will convert into what? Memory B cells. Achha, why the memory B cells? Because next time whenever the same antigen will encounter, so much of recognition, this and that will not be there. Directly it will be attacked in attack mode. Okay, so time will be saved and the patient will survive. But in this case, what do you call, one more type of cell will be originated, which is called plasma cells.
Sir, that is also called as killer cells.
Not exactly. I'll tell you. The TC cells are sometimes known about natural killer cells. Okay, but there is a bit difference. I'll come back to that. So what exactly happens? The B lymphocyte has been primed and therefore it has been converted into memory B cells, which will be helpful for the vaccination, this and that. But the effector cells, memory is one, and then the effector cells is the plasma cells. Now, what is the thing that has motivated the unprimed B lymphocyte to get converted into two different types of cells? So what exactly has happened? One mitosis has taken place here, no? One round of mitosis has taken place, and therefore one cell has broken down into two. So how exactly this whole thing has initiated? There should be some switching on, switching off device, isn't it? So the antigen that has entered into the body has initiated the response, but there are some other things that are needed. What is needed in order to break down into one more type of cell? That is called cytokine, a protein which is responsible for breakdown of the cells into two different type of cells. Understood? Now who will produce the cytokine? Okay, the cytokine is needed so that the unprimed B lymphocyte, close it. The unprimed B lymphocyte